I'll tell you a little bit about my background. I was born in the city of Dhanbad in the state of Jharkhand, and I come from a coal mining family where both my mother's side and the father's side, the entire family were working in coal mines. So I grew up in different parts of the country. Um, Dhanbad for the first 10 years, and then I was in Chandrapur district in Maharashtra, where there are also coal mines. Then I was in Nagpur, I was in Delhi for my 11th and 12th. And then I joined IISER Pune as an undergrad. I majored in biology, and at IISER Pune, I worked with Dr. Mayurika Lahiri for my master's thesis. And post my IISER time, I joined the University of Michigan in United States for my PhD's work. And after my PhD, right now I am in Columbia University, New York City, where I'm continuing my postdoctoral training in the laboratory of Dr. Oliver Hobart. And my employer is Howard Hughes Medical Institute um, that funds
Cool.
different labs across the country, including our lab that is housed in Columbia University's main campus. Stress can come in different forms. Uh, I think in modern society, stress is mostly psychological stress, emotional stress, but then there are also other ancient forms of stress, such as nutritional stress, food stress. Either you have too little food or you have too much high calorie diet and your body is not able to adjust to that level of calories. My current research is about understanding how the brain responds to stress. Specifically, I'm looking at how nutritional stress that is starvation, affects the behavior of the animal. And for this, I use a very simple organism that is called C.elegans or C.elegans that gives several advantages uh, in terms of it is a completely transparent animal. So we can see what is happening inside its body when we subject the animal to stress. We can see how the neurons in the brain of the animal are changing their activity. We can change the differences in the behavior of the animal in terms of how it is moving, how much it is eating, and several other behavior in terms of its reproductive behavior and other features as well. I'm specifically focused on understanding how the feeding behavior is altered when the animal is starved and what controls the feeding behavior of this animal. And I'm focusing on one part of the nervous system that is a very less studied part of the nervous system. This is the nervous system that is in the intestine or the gut of the animal. And all
Okay.
organisms have two brains. There is one brain that is in the head that everyone knows about, but we also have a second brain. In human, this brain has 150 million neurons, which is 1 crore 50 lakh neurons. And these neurons are in the intestine, and their job is to move food from the anterior part to the posterior part, which means from the mouth to the end of the anus of the animal. And this second brain can function completely independently from the first brain. So this is something that I'm studying. How does the output from this independently working second brain change when the animal is under stress? And this has immense clinical application as well, because it has been shown in recent clinical studies that 40% of the world's population suffers from some sort of stress induced disorder of the stomach. So this can be either constipation or diarrhea, what we call as loose motion, and also other forms of diseases. One of them is called IBS or irritable bowel syndrome. So these are different types of stress-induced gut disorders that we don't understand. And I hope that my work with C. elegans will help us understand how the second brain responds to stressful environments. So the connecting factor in all my research experience has been the factor of stress, because I've always found it very fascinating how organisms have evolved to not just deal with the favorable conditions, but also extreme challenges in their environment. So it started when I was an undergraduate BS-MS student in IISER Pune, and I was very fascinated by the disease of cancer, which was because cancer is caused by several factors in the environment that are stress inducing, either at the DNA level or at other levels. So my master's thesis in Dr. Mayurika Lahiri's lab focused on studying these DNA damaging compounds and studying how does this disrupt the tissue structure of uh, the human breast epithelial cells to cause breast cancer, and to study the initial steps of what molecular changes happen when these DNA damaging agents cause genome level stress in these breast epithelial cells. So that was my master's thesis. And during this time, I got interested in studying stress at the level of the whole organism, and more so in terms of the regulation of the entire lifespan of the animal, or what we also call as aging. So, because there are many theories of why we age, and one of them is that we age because our ability to fight stressful conditions goes bad during aging. So, I decided to study aging, and then, as I mentioned, my master's thesis work was on cancer cells using human cells grown in lab conditions. And for my PhD work, I joined the lab of Dr. Alan Shu at the University of Michigan, where I was looking to find a model that is, by model, I mean an experimental organism that is suitable for studying the process of aging. And that is when I first started working on the worm C.elegans or C.elegans, because this animal lives for only about 3 weeks. So you can see the entire process of it reaching adulthood and then slowly aging. And in terms of multiple features, over these 3 weeks, these animals' movement will slow down. These animals will, the eating, the amount it is eating will slow down with time. Its muscle activity will slow down. So a lot of processes that we see um, in regular aging in another any organism is also observed in C. elegans, and it gives us the ability to make many manipulations. Either you can do genetic manipulations where you can modify the function of genes. You can do environmental manipulation where you can change the environment, you can change the diet, you can change the temperature, you can change the humidity, you can change other factors in the environment, or you can take a pharmacological approach where you give the animal some sort of a medicine or a drug and then see if any of these three types of manipulations, environmental, genetic, or pharmacological, can change the lifespan of the animal. It can make it live longer. And my project specifically focuses on the pathways that are involved in fighting stress. And if you can increase the activity of these pathways, can the animal live longer because it has better ability to fight stress in old age? And during this period of my PhD's work, I realized that the brain plays a very important role in detecting stress. So I'll give you an example. This work was done uh, a little bit before my PhD started. They were trying to study the effect of high calorie diet on the lifespan. And what they observed is that if you feed animals high calorie food, they live shorter, which is expected because high calorie diet is not good for you. But if you do what is called as dietary restriction, that is eating about 70% of your maximum capacity, these animals were living longer. Now, what they did is, so again, more food, animals live shorter, less food, animal lives longer. Of course, it cannot be too less because then the animal starves. So it is about 70% the maximum capacity of the animal. What they showed that if you grow the animal in a low nutrient condition, but you provide high nutrient food high nutrition food across a barrier. So it can smell that food, but it cannot eat it. So that is enough. The smell of the food is enough to cause the damage because your brain is making your body feel that you are eating high calories. So this was really interesting. So this got me really interested in understanding the neuroscience aspect of things. How does the brain respond to stress? Because as we mentioned, we all know what will make us live longer and healthier. We need to exercise, we need to eat less, we need to eat healthy, but it is very difficult with all the factors that are available in our environment. So can we trick our brain to believe that we are in a healthy environment? And for that, we need to understand how our brain responds to things. And if we can activate that part of the brain, we can get the benefit without actually doing the thing. So that triggered my interest in understanding the neuroscience of stress. And hence, I joined my current postdoctoral lab, where I'm studying how the nervous system or the brain of the animal, specifically the second brain, as I mentioned in the previous question, deals with stressful environment. So honestly, as I mentioned, nobody in my family comes from a scientific background. So it was a dream to become a scientist, but then I had no role models while growing up. Uh, I the role models were the people, the scientists I was reading about in my textbooks, but I had no idea how to become one. I just knew that I have to study science. So what happened is ICERs were very new back then. I was the second batch of IISER Pune. So it was still a new concept, but when I heard about it, that the government of India is starting an institute solely for the purpose of training undergraduates in basic research so that they can become scientists. This was the best timing ever for this. So I was in class 11th at that point, and I contacted the director of IISER Pune at that point, mentioning my interest, and they told me that there will be um, some sort of a selection procedure, because back then it was not clear if they will have a separate entrance exam or will it just be through the IIT JEE entrance exam. So that thing was decided in a span of time, and then eventually I decided to, even though I had the rank to get into both the good medical colleges as well as good engineering colleges, I decided to join IISER because this was my dream to get exposed to research during my undergraduate training. Now, how IISER Pune helped me in that journey? It helped me throughout. I think the best aspect was that the courses were not based on rote memorization, but they were based on interactive learning. So there were a lot of discussions. There was a lot of questioning existing concepts that makes you develop into better scientists. That was indeed the best part of my education, that we had to think what is in the book, but also beyond books, that what are the questions that for which we don't know the answers to. And secondly, as I feel that another aspect was, since we started working in the lab, we also started reading papers and we started critiquing research papers, that, okay, is this paper good or is this paper bad? And this helped us distinguish between good science and bad science. And this distinction is very important for somebody to grow as a scientist, because when we see a research paper, we don't know the background of it. Uh, but when we read the research paper and we start asking that, was this the right experiment to do to answer this question? Or could you have done this experiment slightly differently? Or could you have done an additional experiment on top of this experiment to come to that conclusion? So these discussions made us better scientists because we were trying to find how to do good science. So yeah, these were clearly the main factors that contributed to my scientific journey. Of course, there was a lot of formal learning, but the informal part, the part where I could work in a lab so early in my undergraduate degree, I started working in a lab right from my third year of undergraduate. That's when I started working full time. That itself was a great privilege because very few undergraduate science students in India have that luxury of working in a lab full time on an everyday basis while they are taking their courses. So I'm really thankful that IISER Pune helped me in that journey. If I'm not wrong, you are trying to ask if joining IISER at that stage, where it was not an established name in
Right.
terms of scientific research, uh, was it a risk? Of course, it was definitely a risk, and I had a long conversation with my parents about it because I could get into an IIT, I could get into the top medical colleges in the country, and then choosing a place which nobody knew about. In fact, uh, I met this career counselor, not because I had questions, but because I just met them at a train station. And they said that they have never heard of this place and they would not recommend it.
Oh.
They thought it was an agriculture institute or something in Pune. So people have opinions, but I think I feel that this is a very key aspect of science. To be a successful scientist, you have to take risk before others. If you have to see where opportunities are opening up, and only then you can take those opportunities and do things that others have not done yet, so you can be ahead of the curve. So I think not just me, but every single person who joined in my year and also the year before me, who even took a bigger risk because they were the first batch in that case. But because this was a government central government institution. So clearly, the idea was to create a world class institution. The name had Indian Institute in it. So clearly, there was going to be fund. We knew that there will be funding. We knew that there will be a focus on developing quality. So that risk was involved. And for me, the big thing was also getting the bachelor's and master's degree together, because I feel that we have kind of segregated this into bachelor's is to get information and knowledge, and master's is when you apply that knowledge. But ICER provided the opportunity to do both things together because there was no clear distinction between a bachelor's and a master's. I mean, you do your research, you do your courses, you do everything together. And this was great because you could sample a lot of different things before you make the decision. And but once we were in ICER and we saw how things were operating, it gave us the confidence that we made the right decision. We were in a place where there was a lot to learn. There was a ample opportunity to grow as a scientist, and at that point, clearly, very few places in the country offered that. In fact, in the US, when I explain the ICER course structure and the ICER design to people, they also feel that this is a fantastic opportunity where you can get a small group of undergraduates and then expose them to research labs so early in their career, and they feel that this is a fantastic model that should exist elsewhere. And since then, we know that this dual degree things have become much more common in India and as well as abroad. So ICER was truly a pioneer in that regard, I would say. I have many memories of ICER because I was there in the initial years of ICER Pune. So there were a lot of factors that we saw change. When we started, ICER Pune was just a one building institute housed inside NCL Innovation Park. And then it grew, it moved to a, temporary campus in Pashan Circle. And then it finally moved to its own campus. During this time, we were also moving hostels. So we lived in an apartment building initially in Sutarwadi because the campus was not ready. So this was the only building that was available to be used as a hostel. And then after that, we moved to the campus and we moved to another hostel that was in the NCL campus. Everything was under construction, everything was constantly changing. I also used to participate in a lot of non-scientific activities, playing cricket with friends in the IISER campus, participating in the drama club, and any other cultural events that were happening on campus. So I have great memories from this. And the friends I made in IISER Pune, I'm in touch with most of them. And I'm really glad that we got to connect over an extended period of time, because in some parts, some courses the undergraduate degree can be for 3 years or 4 years, but ours was for 5 years. So it was an extended period of time to know people um, really well. And I'm glad I have made those friendships and also those bonds with professors, because once a professor knows you for that many years, you have taken multiple courses, they know your strengths, they know your weaknesses, they point out what are the things you can work on. So it really helps build bonds. And when I talk to these professors now, I feel I'm not talking to just a colleague, but to someone who deeply cares about me, because they have seen me grow from a 16-year-old teenager to a more mature person. And they feel very happy because they have been part of this journey. I have joined the alumni portal. I have seen some people from my year and I have connected with them. But I'm in touch with most of them independently over the years via email. But I think this is a great initiative, because there are some people who I would like to connect to, but over the years I have lost touch with them. So this is also a great opportunity for people to ask questions to IISER alumni who have gone in a certain career path, ended up in a certain university, in a certain company, where some current IISER student might be interested in that. And we should definitely use this alumni network to connect these people so that they can reach out with questions and get selective feedback, useful feedback that can help them succeed in their careers. So I think this is a great initiative, and we need to grow this now that IISER alumni have been the first batch graduated in 2011. So it is about 13 years since then. They have all been, they are all in successful positions in life. So they can offer a lot of advice. They can offer a lot of expertise and guidance that will be available to the current students of IISER.